Hello and welcome, everyone. This is a big day for us at Enhanced 3D Genomics. We're finally launching our virtual seminar series, something I've been wanting to do for a long time. And I'm really thrilled to say that it's finally happening. It's actually happening today, right now. So the aim of this series is to hear from leaders in the field of enhancer biology and 3D genome organization about their latest research and findings, and then followed by discussion, hopefully with as many of you as possible. A few housekeeping tasks before we start. So this talk will be recorded. Pradeep has agreed to that. We'll stop the recording before the questions, though, right? So don't worry there. And we ask that you would ask your questions not during the talk. In my experience, that doesn't work so well for Zoom talks, but at the end, either by putting the questions in the chat box or raising your virtual hand. And I think that's what I needed to cover. So yeah, the session is 1 hour long. So talk will be around 45 minutes, 45 to 50 minutes, so that we have 10 to 15 minutes for discussion. Yeah, so with that covered, I'm delighted to welcome Pradeepa Madapura as our first speaker today. Pradeep has done his PhD in Bangalore, in his native India, and then moved to the UK in 2009 to Wendy Bigmore's lab in Edinburgh, and he's done some fantastic work there on non-canonical enhancers. That is also the first time that I became aware with Pradeep's work. And then from there, from Wendy Bigmore's lab, he moved to his first independent position in 2016 at the University of Essex in Colchester. And 2 years later, he moved to Queen Mary University, where he still is today, now as a reader in genome biology. Yeah, and I'm delighted to welcome Pradeep here. Stage is yours.
Thank you, Stefan. Thank you very much for the invitation. I'm going to share my screen and going to private. Yeah, hope you all can see and hear me well. So yeah, I'm Pradeepa Madapura. I'm academic at Queen Mary in London. That's Blizzard Institute, the picture which you can see behind. That's where I work. So I'm part of this Queen Mary Epigenetic Center. We have a really nice group of epigenetic researchers. It's great to present my work today. So I'm going to give quick introduction about the genome and how our DNA is packaged. So you must have heard about all our DNA is packaged with the histone octamers. They basically it wraps around the histones, and then and this kind of chromatin which is packaged with the histone can be regulated by various chromatin modifications and chromatin proteins factors and DNA modification. They play a key role in packaging the DNA. For example, as you can see in the left side, as a dense heterochromatin where there's a lot of DNA methylation and there's irrepressive chromatin modification, or euchromatin or open chromatin where there's a chance for DNA to be accessible by transcription factors and coactivators, and they're mostly marked by some of the chromatin modifications which are associated with the active transcription, or it also allows other DNA mediated processes like DNA repair, etc. Okay, these modifications they are reversible, they change, and then that basically keeps our DNA accessible whenever it is needed for our nuclear processes. So the modifications are obviously too many on the chromatin. So now this is the kind of illustration from AppCam where you can see the histone H3 which is here, and histone H4, H2A and H2B. They all have a histone tail, and in these tail residues there are many lysine, arginine, serine, threonine. They all have potential to get post-translationally modified by acetylation, methylation, ubiquitination, many acylations as well. And there are many enzymes which does these modifications, and there are proteins which recognize these modifications. There are reader domains, and also there are enzymes which remove these modifications. So it's great, and there's so much complexity is known, and of course they all known to demonstrate it to show one or the other function on the chromatin. Some of them, as I mentioned, they could contribute to transcription or chromatin accessibility or function of enhancers and promoters. Okay, so I'm highlighting a couple of here modifications here. That's a lysine 27 of histone H3, and this is the lysine 16 of histone H4. Also come a few other modifications later. So most of my talk, basically, I'll be talking more about this lysine 16, which is on the histone H4 and tail. Right. So not just the stone tails which undergo these modifications. We know that there are residues on the stone H3, particularly there are lysines which are known to be acetylated, for example, at lysine 64, lysine 56, and lysine 122. So these modifications, and also 79 as well. So these modifications are kind of known to have a very different function, not just by attracting the effector molecules, which I mentioned, the reader domains. These molecules these modifications can perturb the nucleosomal structure by altering the interaction between this DNA, which is kind of wrapped around this histone octamer, because these modifications basically neutralizes the basic charge of these lysine residues of histones, and which are important for interacting with the electrostatic interaction of DNA, which is negatively charged. So that kind of charge-based interaction gets perturbed and destabilizes the nucleosome. So why do we need these? They are still, although it's kind of assumed that they are buried, this domain is buried, it's not really buried, it's in the lateral surface of the histone octamer. They're outside, they're still reachable by enzymatic complexes. So these modifications can have functions, for example, altering the nucleosomal stability at the regulatory elements like enhancers and promoters. They can also play a key role in destabilizing and also turnover of the nucleosomes. So what are their function? They could be important for kind of keeping the chromatin or the enhancers and promoters in a poised state where they need to be activated as quickly upon differentiation signaling. Okay, and so this is just an illustration which kind of my lab focuses on, for example. So we know that lysine 27 acetylation is one of the very well studied acetyl mark, which is kind of widely used to identify active enhancers in the genome. We know that many of these acetylations, they are recognized by bromodomain containing proteins, including BRD4. That's again very well studied protein, which is implicated in enhancer and super enhancer function and various other functions in the nucleus. The interesting thing is like all these acetylations, or most of these acetylations, are mediated by one major acetyl transferase complex. That's a P300 CBP proteins, and they acetylate not only K27, many lysine residues on histone H3 tail and some of the H4 tail lysines. And this enzyme also can acetylate globular domains. Particularly, it's interesting because this enzyme can acetylate when it cannot acetylate lysine 27 if this modification is this lysine is methylated, for example. So what's the consequence of this acetylation? As I mentioned, this can perturb the interaction and allow the DNA to be accessible by transcription factors. So that can have a completely independent function of acetylation rather than requiring adapter like BRD4, effector like BRD4s. Lysine 16, as I mentioned, which very interesting because this lysine tail, this N-terminal tail of this histone H4 can interact with the acidic patch of the neighboring nucleosome, and this tail to nucleosome interaction is important for packaging chromatin, higher order chromatin packaging. And this acetylation again neutralizes this charge and then perturb this interaction between two nucleosomes, and can, at least in vitro, can perturb the higher order chromatin structure. So, our lab is kind of interested in understanding some of these modifications, what are their role and where they are present in a genome, what are their how we can use these modifications to understand the genome regulation. So just to give a quick introduction about the genome and gene regulatory elements. So we know these are the genes, for example, the 1, 2, 3, or the 4 numbers. They are different genes. Some of them are basically inactive. They don't have many of these chromatin modifications, and they don't have accessible chromatin there. And some are basically like poised for activation. They are not active, but they have features which kind of guide us, like, they could be active upon differentiation or any kind of signaling. They have these modifications like lysine 4 trimethylation, for example. And once they get an activation signal, they accumulate acetylations, including K27 acetylation. And then we can kind of classify gene transcription start site, a promoter with these combination of modifications are active genes. Of course, most of these have accessible chromatin, so their chromatin accessibility is not great for identifying precisely the activity of the regulatory elements or enhancer or promoter. But I think the using combination of chromatin modifications are the key. So going back to enhancers. So the enhancers again exist in 2-3 different configurations. There are inactive enhancers, for example. They basically don't have any of these modifications, and these enhancers, when they get particular modification like K4 monomethylation, we kind of classify it as a poised or primed, and that again further can be subdivided between poised or primed, depending on whether they have a particular modification, that's a K27 methylation along with K4 monomethylation. This combination kind of predict these enhancers are poised to get activated. And if we don't have this K27 methylation, and if you have just K4 monomethylation, they are inactive enhancers or primed enhancer. They can also get activated upon binding of particular transcriptional coactivator. And of course, these are the enhancers and promoters, and the active enhancers, as I mentioned, like, they basically acquire this acetylation signal, and not just K27 acetylation. I'll come to it next slide. So they also have many coactivators or architectural proteins. They can help in kind of looping to the promoters or genes with the help of these chromatin architectural proteins. So, and of course, all these kind of genomic features, they exist in a higher order chromatin structure. That you can call them as a topological associated domains or TADs. So, so about 15 years ago, I think, when we are trying to use all these genomic methodologies or chromatin modification profiling, we found that most active promoters have this feature of like K4 trimethylation and K27 acetylation. And also most of these enhancers, when they're active, they have this combination of K27 acetylation and K4 monomethylation. And all these are modified by this P300 CBP complex and MLL complexes. Okay, and then over the years, we and other labs have shown that not just K27 acetylation, these enhancers also have many other acetylation, including globular domain acetylation, which I mentioned, that's H3K122. And they also have a K9 acetylation, case H3K14 acetylation, and recently the H2B N-terminal acetylation. They're basically active enhancers have a lot of acetylation. Active genes have plenty of acetylations as well. So it's hard to basically the enhancers promoter look alike, except that the enhancers have low K4 trimethylation and higher K4 monomethylation. About 10 years ago, we showed that although there are all these enhancers have this K27 acetylation and many others of these acetylations. One particular acetylation were kind of unique. That's a H4K16 acetylation, which was previously thought to be kind of marked at the gene body. We showed that actually this marks the active enhancers, and this modification is mediated by a completely different family of acetyltransferase. That's a missed family of acetyltransferase. It's very unique. It only acetylates lysine 16, this complex. So it's very interesting. Like, this can be how it is particularly specifically targeted to independent enhancers which lack this K27 acetylation. We don't know. So we think it's a combination of transcription factors. So that gave us an idea, like, how independent coactivator complexes can bind or recognize genomic features and activate genes independent of this canonical enhancers, which are marked by various other oscillations mediated by P300. So why do we care? I think, as I mentioned, the genome is complex. There many features play a vital role in precise gene regulation, and many of these genes and enhancers are poised or primed for activation. And these chromatin feature modifications are important in identifying the status of these genomic features. So something which kind of feel ignored over the years is the transposable elements, mainly due to technical difficulties and also the complex nature of studying them, because they are repetitive nature. And then we basically threw out all this sequencing data when we were analyzing in the olden days, and then we only focused on the genes, and we also focused on the for example, enhancers and introns, which are constitute nearly this for 50% of the genome, and rest 50% is actually transposable elements. Just to give a couple of examples, these are line 1 transposable element. They constitute nearly 19% in human reference genome. They are about 500,000 line 1 elements. Many of them are truncated, but there are about 10,000 elements, and each of them are about 6, 7, 8 kb long. That's a lot of line 1 elements in our genome, but only about 100 are capable of retrotransposition. There are about 1 million alusine elements. They are smaller, but there are plenty. And also nearly 500,000 ERV-derived sequences. Many of them are solo LTRs. Basically, they are like a transcription factor binding sites, and they are basically deleted. Like we have truncated many of these internal regions, and some do retain internal regions still. So why do we study them? The reason is they all still maintain some of these transcription factor binding site. This is like a promoter of line 1 elements. These are LTRs of these HERVs. They basically bind transcription factors. They can influence the regulatory landscape of our genome because they are so abundant. So, so as I said, the previous thought was like most of these could have a mutational effect, particularly when they're retrotransposed to the gene of interest, they can cause mutations. So that can be prevented by repressing the transcription of these transposable elements by DNA methylation or chromatin modifying complexes. However, there are some transposable elements. They can still escape this repression, and they kind of co-opted or co-evolved to function as a non-coding RNAs, or they can also function as enhancers like elements, and also they could function as an alternate promoter, particularly if they are near the promoter elements, or if they are present within the enhancers, or they could also create a de novo enhancer elements to regulate expression of genes in cis. So obviously that because they have moved so much, they have expanded our genome. They also inserted into the introns, and that created a lot of complexity to our genes. And so that's obviously kind of hypothesized to be important for evolution of our genome. So so now I'm going back to the lysine 16 acetylation, because most of my talk kind of covers this acetylation. This is a very well studied modification in Drosophila. It's known for dosage compensation, male specific upregulation of X chromosome to balance the dosage of 2X in female. But in mammals or vertebrates, we don't really have a same kind of dosage compensation. We have a X inactivation in female. But however, if you look at the complexes, which does the K16 acetylation, that's the MSL complex is kind of conserved from Drosophila to human, so it's the same proteins in both. Very interestingly, that's an enzyme that cat8, if it's in NSL complex, can acetylate lysines 8, 5, or 12 for histone H4. Only when it's present in the MSL complex can acetylate lysine 16. So we have a very interesting complex which does this modification only when it interacts with these proteins. However, the very recent study, when we were trying to understand this function in our lab when I started, there are two papers which came up with the conclusion that if you deplete K16 acetylation by knocking out this complex, there is no major change in gene expression, like if you look at the overall gene expression. So that took us back, like, we started wondering what's happening. So we were studying in human embryonic stem cells, and we are trying to map how this what's the function of these modifications. So when we perform cut and tag for these modifications, along with many other known popular chromatin modifications which are known to be enriched at the promoters and enhancers, for example, K27 acetylation, K4 tri mono, and K122 acetylation, which we have previously shown to be enriched at the enhancers. So when we did that, so we could see that it's kind of known previously K16 acetylation doesn't really enrich at the promoter. It's more on the gene body. That's what we see, while others are in the promoters. When we analyze carefully, like, we found a lot of this K16 acetylation is going to these transposable elements. That's the line 1 elements, which is kind of supposed to be repressed or enriched with the K9 methylation. So going back to line 1 structure, for example, this is line 1. It's got a 5' UTR, which acts as like a promoters to transcribe the full length line 1. And these are open reading frames, and which encodes for proteins important for reverse transcription, etc. So we found very specific enrichment of this K16 at the promoter-like element of these line 1s. And also we could see enrichment for K122, and then along with K4 monomethylation, very little K4 trimethylation compared to these promoters, and then a bit of K9 methylation in some of these line 1s, and low level of K27 acetylation. So that kind of took us surprise. Why do we see this K16 on these line 1 elements? So when we analyze the data carefully, we found very interesting, like, a lot of these line 1s appears to loop with the genes. When I say loop, basically they make a 3D contact with the genes in cis, particularly when they are acetylated. If you compare the non-acetylated versions of this transposable element, this is a non-acetylation. Any of these acetylation is making them loop to the genes, make a 3D contact on all three different class of transposable elements which we studied. So we wanted to study this further. Like, it's we are curious, why do they have these lines loops to the genes? And then we took approach of CRISPR-Cas9 to and then dCas9 to perturb these transposable elements and also delete these transposable elements and see their effect on cis regulation. So this is an example of a transposable element. We deleted this. This is a long gene of about nearly 100 kb, and we could see that the transposable element region interacts with the promoter of this gene and also the genes nearby, but not this side, this gene. So we could specifically perturb this transposable element, and we could see the effect only on these two genes, but not this gene. So this data is published. I'm not really going in detail, so it's you can read it. So moreover, what we could do is, like, we can perturb this complex by, you can deplete the complex and then see that effect. When we deplete, we lose the K16 acetylation, and also what we see is like reduced transcription of this many of these transposable element which are marked by this K16 acetylation. Our working model is like we have transposable element all over our genome. They are at the enhancer, they are at the near the promoters, they could be on the introns, or they could be intergenic regions. And most of these transposable elements in somatic tissues, they are repressed by DNA methylation and kainine methylation, and Krabs finger and Asch complexes, for example. And while some of the other transposable elements kind of escape, or they evolve to function a tissue-specific role in functioning as active enhancers to regulate gene synthesis. Also, many of these transposable elements could have a tissue-specific role. The transcription itself might have a tissue-specific function. So we show that this complex regulates transcription of this element and also can influence the expression of genes nearby gene synthesis. Okay. So we showed that basically, sorry, that's not supposed to be there. Yeah, so we showed this modification plays a key role. Sorry, I don't know what happened to this. It's going back to, it skipped the page. It went back to, okay, yeah, so this is where I want to be. I don't know. What we show now is like, the histone acetylations are associated with active genes and enhancers. And it's not just K27 acetylation which marks which distinguish active enhancers and genes. We also have a few other modifications. If you look carefully, we can distinguish them, the poised or active enhancers. Particularly the lysine 122 and K16 acetylation can also identify regulatory elements. And we showed this line ones and ERB LTRs marked with acetylations can function as enhancers. Of course, the LTRs were previously shown, but we are one of the first group to show the line ones can indeed function as enhancers. And also this complex can activate the transcription of line 1 elements. So these transposable elements not only expressed in embryonic stem cells, which I've just shown my data, they're also expressed in trophoblast lineage and also in neuronal lineage. I'm not presenting any neuronal data here, but I'm going to go through a rest of my talk on the trophoblast lineage. Basically, the trophoblast stem cells make most of this placenta. And the placental trophoblast stem cells in placenta are co-opted in many of the transposable elements to have the trophoblast specific function. For example, this is the gene syncytin, which is from ERV, and which functions as a kind of a is envelope derived gene. It is important for this kind of a syncytia trophoblast fusion of cell fusion function. And many of these transposable elements in trophoblast also can function as a placental specific or trophoblast specific enhancers. So rest of my talk, I'm going to talk about particular disease condition. It's called preeclampsia. So this occurs due to reduced trophoblast invasion. For example, when during placentation, this extravillous trophoblast, which is derived from trophoblast stem cells, they're supposed to invade into maternal decidua, and then leading to remodeling of these arteries, and which allows for the exchange of nutrients. And so when this kind of reduced invasion of this trophoblast due to altered differentiation or invasion can cause a condition called preeclampsia. It's basically hypertension associated with pregnancy, which occurs very late in the pregnancy, around 30 weeks of gestation. But most of this placentation defect it occurs very early on in the first trimester itself. But so far there is no approved test to predict who is going to end up developing this preeclampsia. Of course, it's a big burden for the healthcare system, including NHS. If you want to know, I recommend watching this BBC TV drama. It kind of demonstrates how much the burden it can cause to NHS because of the extra care and which occurs due to preterm birth. So um, so I'm going to talk about what we are doing so far now with preeclampsia. So in a collaboration with Ajay Sinha, and we are a MD PhD student from Egypt. So we first aim to profile epigenetic changes in preeclamptic placenta, and we found very interesting hyperacetylation of lysine 16 and also some hyperacetylation of lysine 27 in preeclampsia specific placenta, but not less in control, but more on the preeclampsia. And it's multiple placenta, which we are just averaging it. What was interesting is like consistent with our previous finding, a lot of this hyperacetylation is happening at the line 1 elements, particularly lysine 16 hyperacetylation, while the K27 acetylation is happening all over, like, all the genes and transposable elements. So we are curious why we see the hyperacetylation in preeclampsia. And when you look at the transcriptomic data, our own data, and also some of the published data sets, we could see a lot of changes in metabolic pathways, including glycolysis and the pathways which basically alters the acetyl-CoA level due to altered metabolism. It's also known that preeclampsia is associated with premature aging and also elevated senescence marker. There's a lot of links to K16 acetylation with aging and senescence. So we basically thought we need to characterize this further. So we got hold of multiple cohorts of transcriptomic data set from placenta, from preeclampsia and controls. So this is a complex heat map. I'm just quickly going to explain what it is. If you look at this group, basically that's red is a preeclampsia and blue is a control. If you look at these red groups, that's a preeclamptic samples. They all have upregulation of transposable elements, very specific to preeclampsia. Compared to the blue ones, they are control samples. And there are few are down-regulated in preeclampsia, but it's the upregulation appears to be a lot more stronger and very specific. It doesn't matter what type of preeclampsia. Preeclampsia could be early onset, or preeclampsia along with the intrauterine growth restriction, or severe preeclampsia, depending on how you classify it. It appears that they all have a higher level of transposable element. And we also looked into some of these data set which are kind of gestational age matched. And we could see consistent result, like, it's not due to altered early delivery of the placenta. It's all age matched placenta. So what happens were these transposable elements or viral elements in the end, viral DNA, they have been integrated in our genome. We know that when we are infected with virus, we host response with the type 1 interferon response using this structure recognition proteins like RIG1, MDA, and proteins. And same thing happens when we upregulate transposable element, whatever the reason, whether you perturb the epigenetic machinery or natural causes due to stress or aging or any conditions. So they are sensed by the cGasting pathway, and that could lead to type 1 interferon response. So that's what we see. Like, when we analyze the data set, we could see increased transposable elements in preeclampsia, leading to higher level of type 1 interferon response. As you can see, the one which are upregulated in preeclampsia, they all have a type 1 signature. So we know that preeclampsia has got a high level of inflammation. So we think the transposable element deregulation could be one of the contributor for this inflammation we find in preeclampsia. So so I asked my postdoc at that time, so because we are seeing a very nice signature, we see a upregulation of transposable element in placenta with preeclampsia. Placenta is a fast growing organ. It's a rapidly changing, and it's a large organ as well. So what happens when there are a lot of these organs in our tissue, they always shed nucleic acid there, because when the cell undergo necrosis or apoptosis, all the DNA comes into the circulation. So for example, if you take a if you just take a blood from a pregnant mother, you can see the DNA coming from various maternal tissues, from brain, heart tissue, brain, heart, liver, also fetal cells, and also placental cells. So there is a proof of principle study by Illumina. They showed that by using a liquid biopsy approach, by taking a blood from a pregnant woman, we can kind of see that there's a large contribution of fetal and placental or fetoplacental DNA in the maternal blood. So that same concept has been used by researchers in Stanford. They basically took a cohort of nearly 400 pregnancies, blood samples, and then at the early stage, at the first trimester of the pregnancy, for example, that's the stage when the preeclampsia onset happens, but we don't see the symptom. The symptoms only occurs at the third semester, but there is no way we can predict, there's no current methodology to predict who is going to have preeclampsia. So the idea was like, if we take a blood from a mother with, when they come for a first hospital visit, and then profile the transcriptome, can we predict who might end up having a preeclampsia? So they showed this can work. They use the mRNA and non-coding RNA from this circulating RNA seq data sets, and they could predict the preeclampsia. And then because we see a really strong transposable element deregulation, we wanted to do the same thing with the same data set. So we basically analyzed the data again, and they found a lot number of transposable elements are up and some are down-regulated in this preeclampsia circulating RNA. And then again, we could basically separate preeclampsia from controls. Again, the red is preeclampsia, blue is controls. So this is even before the symptom appears. It's not even from a placenta. It's from a blood of a mothers who was given blood sample for the in the cohort study, and we could see a upregulation of many of these transposable element or higher level of transposable elements in the maternal plasma. So we basically used that approach, and we wanted to see, is it actually coming from a placenta? Is it actually coming from trophoblast stem cells? Because as I mentioned before, there will be signature coming from maternal tissues, maternal organs. Also, there will be some from a placental or fetal origin. So when we analyze the transposable element, which are deregulated, for example, upregulated at least in this blood plasma, for their expression level in the placenta, using a FACS-sorted placental cell types, very interestingly, we could see that most of the signature which are upregulated they're all coming from this extravilous trophoblast or fibroblast or endothelial cells, while or cytotrophoblast, while the immune cells are not having that kind of upregulation. So compared to immune cells, these ones are upregulated, which gives us an idea, like most of this signature, what we are getting in the plasma, maternal plasma, are coming from the trophoblast lineage. So what we did in my postdoc, what he did is like he used this differentially deregulated transposable element, and then used several filtration to predict or to identify or come up with a machine learning model to predict which transposable element expression signature can be used to predict the preeclampsia. So these are some of the steps we used, and then we came up with 11 transposable element subfamilies. So remember, we are analyzing the subfamilies, not the individual elements, because there are a lot of elements. So we collapsed all the elements into 1,000 subfamilies, nearly 1,180 subfamilies, and then we come up with 11 subfamilies, which can actually predict with very high accuracy who is going to end up having a preeclampsia. So this is the prediction data. The blue is basically a discovery cohort. We trained the model using this cohort, which gives a really good prediction accuracy of 0.94, which is near perfect. And then we use the data set, which is not used for training. that's a validation cohort that from a plasma RNA isolated from around 13 or more weeks pregnancy. We got really, again, very good prediction accuracy. Indeed, it was much better than the mRNA-based prediction of accuracy. And obviously, when we increase the gestational age of the samples, the accuracy further increased, almost as good as the discovery cohort, which again shows that I think the contribution actually is coming from a placenta. More older the placenta is, more bigger it is, and more shredding of nucleic acid, and more stronger signature we are getting it. However, when we also tried very early on, just samples less than 16 weeks of pregnancy, we still get a really good prediction accuracy with we get nearly 74% positive prediction value and 88% negative prediction value. So currently we are at the stage where we are trying to develop this further, and then it can be developed further as a kind of a diagnostic test or which predictive test, which can actually identify who will end up having a preeclampsia very early on in the pregnancy. So yeah, just to summarize the second part. I've shown you that transposable elements are upregulated in lineages like stem cells, in placenta, and also in neuronal lineage, and otherwise they are silenced in the somatic cell types. And the acetylation level, particularly the K16 acetylation level, correlates really well with the higher transposable element expression in preeclamptic placenta. And so we think this acetylation mediated, or there could be other epigenetic contribution as well. This transposable element, which are deregulated, could contribute to higher type 1 interferon response, which is known to, preeclampsia is known to have inflammation. And this T deregulation could be contributing to disease or preeclampsia phenotype. So we have now have a new approach of using transposable element signature in the liquid biopsy to predict or early diagnosis or early prediction of preeclampsia, which is really useful for kind of a prophylaxis. We can start giving aspirin, for example, at the less than 20 weeks of pregnancy. That can change the severity of the disease and also reduce the severity the preterm birth as well. So with that, I'd like to thank my group, which Manthan was a postdoc who's contributed significantly to the first part of the work which I presented, and also is a leading author in the second part as well. Debashri is currently in UCL. She contributed the first part. And I'm not presenting the work of Fanny and Ziba. Adriana contributed to all the project in the lab, and Charlie is also working on following up some of this work. Also, thanks to patient and cohort and hospital collaborations, and some of the basic scientists and also clinicians for their collaboration. And most of the funding in my lab is from MRC and BART charity. Also, we had received small seed funding from Egyptian Council for the MD PhD scholarship. With that, I'd like to thank you, and I'm happy to take any questions.